Greetings students, welcome to DigiNov. I am Dr Gurmanpreet Gill, your faculty for Biochemistry, and today I will be discussing about questions asked on Biochemistry in INI-CET November 2025 exam. So students, how many questions you get from 2025 INI-CET November 25 from Biochemistry is almost 20. So 20 is a good number of questions, so biochemistry is important. Do revise and make concepts about biochemistry to get good marks in INI-CET. So which topics were asked in INI-CET? So most important topic which is asked every year is about genetics. So genetics is asked, around 5 questions were asked from genetics. So which topics from genetics? They were mainly asked about regulation of gene expression and genetic techniques. So these are the 2 topics which were asked, and about 5 questions we get from genetics. Then students, we get almost 4 questions from lipid. So lipid, you should know about metabolism. They have asked about beta oxidation. They have asked about what are the requirements for fatty acid synthesis and what is the process of fatty acid synthesis, especially Carnot and shuttle. Then they have asked about sphingolipidosis, the topic which is asked every year. Then the third important topic which was asked about carbohydrates. So carbohydrates, again they ask about various metabolic pathways like there was question about glycolysis, TCA cycle, HMP shunt, and one question which is repeated every year is from glycogen storage disorders. Then students, we get question from vitamins, we get question from transporters, required primary transporters, secondary transporters, and latest scientists, Nobel Prize winners. So these were the questions. So I must say many questions was from PYQ and PY topics, but some questions were new and they were also not given in your standard books. So better to grasp these topics for next exam, but it was difficult to solve them or in current exam because they were out of your textbooks. But most of the topics was from your PYQs and on the basis of your understanding and concept, and you should be able to answer those questions. But some questions which were new, and so you should leave those questions and prepare those topics for next exam. So now let's discuss various questions which was asked as. So first question was, in rheumatoid arthritis, increased expression of TNF alpha and interleukin 6 in synovial tissue is most likely due to which epigenetic mechanism? So basically students, there were many questions in which your concepts were checked. So this all story was told to confuse you, but basically they ask about epigenetic mechanism, that is methylation and acetylation. So we know that with acetylation, the DNA regulation of gene expression, it is more expressed, more activated. So you should remember that acetylation activates the gene expression, that is more DNA is expressed, more protein is formed, whereas methylation decreases the DNA expression. So less protein is formed, less DNA is expressed. So these 2 things you should remember to answer such question. So first choice was inhibition of DNA methylation. So we know that methylation decreases the DNA expression, that is decreases the gene product, but here it has been increased. So it is correct choice. So inhibition will lead to increase. Second is activation of DNA methylation, whereas methylation is causing inhibition. So it is the wrong choice. Similarly, inhibition of histone acetylation. So we know A for acetylation, A for activation of gene expression, but if it is inhibited, so it will be expressed less. So again it is a wrong choice. Then last choice is activation of histone, deacetylation. No, deacetylation is opposite to acetylation. So if activation of acetylation will increase expression, activation of deacetylation will again decrease expression. So this is also incorrect choice. So the correct answer is inhibition of DNA methylation. So here the concept you should know is about what does acetylation cause? It increases the gene expression, and what does methylation does? It decreases the gene expression. So this question was based on this concept. Next question student was asked about cancer cell, again from regulation of gene expression. Cancer cells show increased RNA transcript length compared to normal cell, most likely due to. So we know that cancer cells have some different DNA. That's why the cells are cancer. They form tumor. So how it is possible? So it is possible due to increased R transcript length. This can be one of the choice. So because of RNA transcript length is changed, the protein formed will be different and it will act differently, which could lead to cancer. So now, moreover, we know that proto-oncogenes are converted to oncogenes, which lead to cancer. So here, but how increased RNA transcript length is possible? By alternative RNA splicing. So what happens when there is post-transcription modification? There can be alternative splicing. So here you can see in protein 1, 2, 4 and 5 and 6 forms the transcript, whereas in protein 2, 1, 3, 5 and 6 form the transcript. So and in last protein 1, 3, 4, 5, 6 forms the protein. So here you can see the protein length is altered. So when protein length is altered, protein formed will be different and it can behave like oncogene. So proto-oncogene can be converted to oncogene because of alternative RNA splicing. So the correct answer is alternative RNA splicing. Why not polyadenylation? So we know that polyadenylation has nothing to do with the gene expression. It is just to stabilize the RNA, mRNA structure. So polyadenylation cannot cause gene to express differently. Similarly, gene amplification. So gene amplification means more formation of same protein. It doesn't mean that more cell division. Cancer occurs due to more cell division. More cells are formed which give rise to tumor, but more protein formation will not lead to cancer. Similarly, gene silencing, again it will not cause cancer because silencing of gene will not be responsible for activation of cell division. So the correct answer is alternative RNA splicing. So the next question, which is important and which I will say is out of syllabus because most of the books it is not given, but here we should know this topic for the next time. So it is an important topic because it is asked. So in this given image, show transporter protein using ATP. So it is a protein using ATP, means primary active protein. For substrate movement across membrane, identify the transporter. So here, because ATP is used for transportation, it means the primary active transporter. So you should know the examples which are examples of primary active transporter, which are secondary active transporter, and which one are facilitated diffusion. So P-glycoprotein directly use ATP for transportation. So it is example of primary active transport. So this is the correct answer. Why not organic anion transporter and sodium glucose co-transporter? Because they are secondary dependent on ATP. They do not require ATP directly for this transport, but they require it indirectly because so they are secondary active transport. Similarly, glucose transporter 4 is example of facilitated diffusion. It does not require ATP, it does not use ATP, so it is the wrong answer. So the correct answer is P-glycoprotein. So for next time, the topic which you should know is what is passive transport where no ATP is required. It can be further simple diffusion, facilitated diffusion as GLUT transporters and osmosis. Then is active transport where ATP is required, which can be primary active transporter which requires directly ATP like sodium potassium pump, calcium ATP pump, P-glycoprotein and CFTR-Cl channels. They directly require ATP, so they are primary active transport and it was asked in question. So the correct answer was P-glycoprotein. Similarly, secondary active transport, they require ATP but not directly. They require ATP for some other reactions which facilitate this reaction also, like SGLT transporter, sodium calcium exchanger and organic anion transporter. So students, you should know about these mechanisms which are primary active, which are secondary active, which are passive transport to answer such questions. Next question was asked about HMP shunt. So here a story was told that a 27-year-old male developed fatigue, jaundice, dark urine after taking antibiotic. That is 2 days after taking antibiotic. Lab test show anemia, raised LDH, elevated indirect bilirubin, negative Coombs test and G6PD deficiency. So we know that in G6PD deficiency are prone to some drugs. So after taking antibiotic, this all happens. So it can be a case of G6PD deficiency. Which statement is correct about HMP shunt? So this all story was told to ask about HMP pathway. So basically they are checking your knowledge about HMP pathway. So in HMP pathway does not produce ATP? Yes, it does not produce ATP. It produces only ribose sugar and NADPH. So this is the importance of HMP pathway. It does not produce ATP. So this is a correct choice. Second, it generates ribose 5-phosphate. Yes, it do. Oxygen, no, and NADPH. So this is again wrong answer because it does not generate oxygen. HMP does not generate oxygen. Third choice is transketolase require thiamine as cofactor. So students, if you remember, for checking the deficiency of thiamine, we do transketolase test because this enzyme require thiamine as its cofactor. So it does not function if thiamine is deficient. So if thiamine is deficiency, transketolase reaction will slow down because it requires thiamine for its activity. So yes, this is true. Transketolase require thiamine. And the last one is non-oxidative phase is irreversible. So if you remember HMP pathway, non-oxidative phase, mainly they can interchange and reverse. So this is again a wrong choice. So the correct answers are 1 and 3. So you should have knowledge and concepts, then you can answer these questions. Next question, again a difficult one, was asked about genetic technique, that is M13 bacteriophage vector used to identify specific gene sequences. Viral spike protein represent which type of expression system? So here they are asking about recombinant DNA technology where a vector is required and it is used to form recombinant DNA, but here it is asked, they are asking examples, which one is example for which. So again, this is a new topic which you should know for next time. So there are different type of expression system, bacteria, yeast, phage and mammalian, and there are various vectors used for these expression system like plasmid vector for bacteria, shuttle vector for yeast, phage vector for phage and mammalian expression plasmid for mammalian expression. So here student, they should know about the example. So it was a difficult question. So PET, PUC and PB are 3 to 2 and PGEX are for bacterial expression. All starting with Y are for yeast expression except PPICZ, and for phage expression as we are asked is M13, lambda and T7. So these are example vectors for phage. Similarly for mammalian it is pcDNA, PCMV, PSV and PEF. So these you should remember for the next time. So this is an important topic. So next question was again on genetic techniques, which of the following represent the correct sequence of steps in PCR. So if you remember, there are 3 steps in PCR which are repeated again and again, and we give millions and billions of copies. So what are these steps? First one is denaturation, in which 2 double-stranded DNA is separated into single strand. Then is annealing, in which primers anneal to these single strand. Then is extension, where DNA is synthesized and 2 daughter copies are formed. Again, they are subjected to denaturation, annealing and extension, and it goes on to form million and billion of copies. So here the correct answer is denaturation, annealing and extension. Do not get confused with choice 1 in which hybridization is given. Hybridization does not occur in PCR. It occurs in blotting technique, where we hybridize probes with the DNA to identify single-stranded DNA to identify them. So it does not occur in PCR. It occurs in blotting technique. Then next student question was, during B cell maturation, expression of single immunoglobulin heavy chain allele ensures antigen specificity. So this is important question, and you must have read it many times, that how it is possible to get so many antibiotics. So our body can produce antibody to almost every antigen. So there are million of antigens, and our body are able to produce antibody against every antigen. But how it is possible? It is possible due to VDJ rearrangement. So we have almost 40 different types of variable segments, 25 diversity segments and 6 type of joining segment. So how many different combination are possible? Are almost 40 into 25 into 6. So many combinations are possible, leading to production of different types of antibodies. So because of this VDGAR rearrangement, our body is able to produce antibodies against many different types of antigens. So the correct answer is VDJ rearrangement. Next question is about glucose tolerance test. So based on the oral glucose tolerance test, which curves are consistent with diabetes? So here students, to answer this, you should know about the ADA criteria for diagnosing of diabetes. So one criteria is the fasting glucose should be more than 126. But here we are talking about glucose tolerance test. So it is not fasting. It is after giving glucose load. So second, after giving glucose load, it should be more than 200 to make the diagnosis of diabetes. So how many curves are above 200? There are two, C and A. So there are two curves which are more than 200 after giving glucose load. So the correct answer is A and C. So here let's revise the ADA criteria. So fasting glucose should be more than 126, and postprandial glucose, that is given after level after checked after giving load, should be more than 200 as we have discussed, and or random sugar should be more than 200. So here, because there are two curves which have more than 200 after giving load, that is postprandial glucose is more than 200. So the correct answer is for choice for curves A and C. Next question you have is to match the amino acid with their respective derivative. So again here you should know about which amino acid give rise to which derivative. So we know tyrosine give rise to thyroid hormones. Yes, correct. Tryptophan give rise to melatonin. Again correct. Glycine give rise to taurine. No, glycine give rise to creatinine. We know glycine, arginine, methionine together forms the creatinine, whereas cysteine forms taurine. So here the A1 is correct. So it is given A and B, both A1 and B2 are correct, but for the C it should be 4. So the correct answer is A1. So here students, you should revise your concepts. Which amino acid give rise to which type of derivative? Then you can answer these questions. Then question was deficiency of which enzyme results in increased 2,3-BPG level and low glucose values in vitro. So this is again a question on glycolysis. So you should know the important enzymes of glycolysis, then only you can answer. So here 2,3-BPG, we know it is formed in a cycle. It is the intermediate step of glycolysis where 1,3-bisphosphoglycerate is diverted towards formation of 2,3-bisphosphoglycerate. So if which enzyme deficiency will lead to increase in 2,3-bisphosphoglycerate? So 1,3-bisphosphoglycerate have two fate. One is it can convert it to 2,3-phosphoglycerate in presence of enzyme phosphoglycerate kinase. Second, it is converted to 2,3-bisphosphoglycerate. So if this enzyme is not working, what will happen? So the 1,3-bisphosphoglycerate will be diverted towards 2,3-bisphosphoglycerate and its level will increase. And glucose, because glucose level decreases because it is used up to form 2,3-BPG. So the correct answer is phosphoglycerate kinase, the enzyme which if deficient will be leading to formation of more 2,3-BPG because it will block one pathway of glycolysis. So the 1,3-bisphosphoglycerate will be accumulated and will be diverted towards 2,3-BPG formation. So the correct answer is phosphoglycerate kinase. So the next is how plasma magnesium levels are primarily maintained through. So magnesium levels in our body are primarily maintained through renal reabsorption. So this is the correct answer is renal reabsorption. So the mechanism which our body follow to maintain plasma magnesium level is renal reabsorption. It reabsorb at the level of kidneys. Then the next question is, this question is a 6-month-old infant present with hepatomegaly, doll-like faces, lactic acidosis and anemia. So there is hepatomegaly, doll-like faces, lactic acidosis, anemia. Liver biopsy reveals hepatocytes packed with glycogen and lipids, suggesting glucose 6-phosphatase deficiency. Which laboratory finding is most characteristic of this disorder? So this is very important and very frequently asked question about glycogen storage disorder, that is glucose 6-phosphatase deficiency leads to von Gerk's disease. So how does von Gog's disease present? So in von Gog's disease, glucose 6-phosphate is not converted to glucose because of deficiency of this enzyme. So basically when we break down glycogen to give glucose, there is a deficiency of enzyme glucose 6-phosphatase. So glucose 6-phosphate is not broken to glucose, which will lead to increased glycogen levels because they are not used. So it is increased, and increased glycogen will lead to hepatomegaly as well as glycogen accumulation as given in question. Then this glucose 6-phosphate can be used to can undergo glycolysis because it is an intermediate of glycolysis. It can form pyruvate, but excess of pyruvate will lead to formation of lactate, resulting in lactic acidosis, and moreover it can lead to more pyruvate can be also give rise to acetyl CoA, which can lead to hyperlipidemia. And this glucose 6-phosphate is also diverted towards HMP shunt because this is also intermediate of HMP shunt, which can lead to more purine formation because ribose 5-phosphate is formed more, which will lead to more purines, and which can lead to more uric acid formation, leading to hyperuricemia. But one thing which occurs, which is most common, is hypoglycemia. Why hypoglycemia? Because whenever we are fasting, we need to break down glycogen to get glucose. But for breaking glycogen, we need glucose 6-phosphatase. And this enzyme is deficient in this patient. So, glucose 6-phosphate is not broken down to glucose, leading to hypoglycemia. So, the correct answer is hypoglycemia, which fasting hypoglycemia, which is the most important characteristic. So, low lactate concentration will be there? No, there will be increase in lactate concentration because glucose 6-phosphate is diverted toward glycolysis, and in glycolysis it converted to pyruvate, which can give rise to lactate. Similarly, elevated cholesterol levels, no, it will lead to increased TG level synthesis. Reduced glutathione levels, no. So, the correct answer is fasting hypoglycemia. Now, next question is, which of the following statements about beta oxidation of fatty acid is incorrect? So, this question we have got these choices. So, different students are telling different choices. So, but let's discuss about this topic so that any choice comes, you can comfortably solve this question about beta oxidation. Basically, this is question asked about carnitine shutter. So, beta oxidation of fatty acid, carnitine is released in the mitochondrial site. Coenzyme A is released in the cytosol during acyl carnitine formation. Both carnitine acyl transferase 1 and 2 transport fatty acid across mitochondrial membrane. ATP is required for fatty acid transport across mitochondrial membrane. So, let's discuss and comes to the conclusion. So, for this beta oxidation, first of all, we know that fatty acids are present in cytoplasm, which need to be transported to mitochondria so that they can undergo beta oxidation and give us energy. So, for fatty acid to enter the mitochondria, first it is to be activated, which require ATP. So, ATP is required to get activated, fatty acyl CoA. Now, this fatty acyl CoA will combine with carnitine in presence of carnitine transporter, palmital transporter 1, transferase 1 to form acyl carnitine. And this acyl carnitine will then enter into mitochondria with help of second protein, that is translocase. So, it is not CPT1 which is transporting protein inside mitochondria, it is translocase. CPT1 is rather converting acyl CoA to acyl carnitine so that it can be transported inside. And inside acyl carnitine again reacts with CPT2 to give carnitine. So, carnitine is generated inside the mitochondria and ATP used, acyl CoA is given out. So, these are acyl CoA is given. So, let's see what are the correct answers. So, carnitine is released in mitochondrial site. Yes, it is released in mitochondrial site because inside it is released. Coenzyme A is released in cytosol during acyl carnitine form. Yes, so we know that in this reaction this acyl carnitine, so acyl CoA is first reacts with that. So, it is released in the cytosol during acyl carnitine formation, it is utilized. Then both carnitine acetyl transporter 1 and 2 transport fatty acid. So, they do not transport fatty acid, rather they helps in transporter, translocase enzyme transport. They only convert it into different products. So, this is incorrect choice. Then ATP is required, yes. For activation of fatty acid, we need ATP. So incorrect choices, carnitine transport 1 and 2 are both transport they helps in transport, but they don't transport themselves. For that, we have a different protein, that is translocase. So these choices, most of the students are confused, so not correctly they remember these choices. But here carnitine palmitoyl transferase 1 and 2 only helps in this shuttle, but for transport we need another protein, that is translocase. So it is a definitely incorrect answer. So next student question was a patient with megaloblastic anemia. So what is megaloblastic anemia? Basically DNA is not formed properly, leading to bigger RBCs. So why DNA is not formed? Because folic acid is not available. It's treated with folate alone, but here when folate was given, neurological symptoms worsen. So what could be, even folic acid is given, this megaloblastic anemia is treated, but neurological symptoms worsen. So what could be the cause? So here students, you should know about the folate trap. What is folate trap? So for folate trap, we need active form of folic acid to be active form of folic acid. To convert folic acid to active form, we need vitamin B12. So if vitamin B12 is not there, folic acid will not be converted to its active form. So again, there will be megaloblastic anemia because folic acid is not available for synthesis of newer RBC, leading to bigger DNA, bigger cells. So here when folic acid is given, active form of folic acid is given by supplement, so folic acid problem is solved, but vitamin B12 problem is still there, and vitamin B12 is required for proper function of nerves. So if we solve the problem of megaloblastic anemia by giving folic acid, but the vitamin B12 levels are still low, so here because they are still low, it will lead to neurological symptoms. So which mechanism explains the phenomena? So masking of vitamin B12 deficiency. So basically here there is not folic acid deficiency, here is a deficiency of vitamin B12, due to which folic acid is not activated, leading to megaloblastic anemia. But when we give folic acid supplement, that problem is solved, but vitamin B12 is still there, so resulting in neurological symptoms. So the correct answer is is a masking of vitamin B12 deficiency, which results in improvement of megaloblastic anemia but worsening of neurological symptoms. Next question asked was, which TCA cycle intermediate acts as precursor for fatty acid synthesis? So here students, the most important is citrate. So this is important. Citrate is transported inside and is converted to acetyl-CoA again. And this acid, you know, the lipids are synthesized from acetyl-CoA. Acetyl-CoA is required for fatty acid synthesis. Moreover, acetyl-CoA is converted to malonyl-CoA in presence of acetyl-CoA carboxylase. So here citrate has two functions. One, citrate inside the cytosol is converted to acetyl-CoA, which is required for fatty acid synthesis. Second, citrate is activator of enzyme, the rate limiting enzyme for fatty acid synthesis, that is acetyl-CoA carboxylase. So it is activator of this enzyme. So citrate is the correct answer, which acts as a precursor of fatty acid synthesis, because citrate is transported in the cytosol where it is converted to acetyl-CoA, and acetyl-CoA is required for the synthesis of fatty acid. Moreover, citrate also activates the enzyme which is rate limiting enzyme for fatty acid synthesis, that is acetyl-CoA carboxylase. So it has two roles in fatty acid synthesis. First, it provides acetyl-CoA which is converted to malonyl-CoA. Second, it activates the enzyme which convert acetyl-CoA to malonyl-CoA. So the answer is citrate. Next question again about sphingolipidosis is, which of the following statement is true regarding Gaucher's disease? So here students, again this is asked many times. So Gaucher's disease is due to enzyme defect beta glucocerebrosidase, and it gives the crumpled tissue paper appearance. So correct choice is B and D. Let's see what are the other options. So here this table is very easy and very important, asked again and again your NEET PG and INI-CET exam. First is about Niemann-Pick disease. So the enzyme deficient you should know is sphingomyelinase. And because this enzyme is deficient, accumulation is about sphingomyelin. So, and it is characterized by foam cells. So here foamy macrophages are also, it is a characteristic of Niemann-Pick cells, not Gaucher's disease. So then is Tay-Sachs disease, where due to deficiency of hexosaminidase A lead to accumulation of GM2 gangliosides. In last exam it was asked, accumulation of GM2 gangliosides occur in Tay-Sachs disease, and here characteristic feature is onion skin lysosome. Then is Gaucher's disease, which is asked this time. It is glucocerebrosidase, lead to accumulation of glucocerebroside and lead to wrinkled paper cell appearance. So these are the very important and very easy table you should remember. So differentiating features are that in Niemann-Pick all the three features, mental retardation, cherry red spot and hepatosplenomegaly is present. Whereas in Tay-Sachs, there's two features that are main features are mental retardation and cherry red spot. Whereas in Gaucher's there is only one main feature, that is hepatosplenomegaly. So if you remember this table, differentiating features, you can easily answer such questions which are asked again and again, sometimes 2 or 3 times in same exam. So here Gaucher's disease is asked, so the correct answer is beta glucocerebrosidase and crumpled tissue paper appearance, whereas Sphingolipidosis and foamy macrophages are seen in Niemann-Pick diseases, whereas for Tay-Sachs disease the enzyme is hexosaminidase and onion peel lysosomes. Next question is, which statement regarding Conn syndrome is incorrect? So Conn syndrome lead to high sodium levels, low potassium levels and metabolic alkalosis. So this you should remember, this is an easy question. So hyponatremia is a wrong choice. Then students, the question comes about Nobel Prize for Physiology or Medicine is awarded for. So here again you should know latest Nobel Prize winners. In 2025 it was Mary E. Brunko and Fred Ramstel. So it was for peripheral immune system which was asked. In 24 it was for microRNA and its role in post-transcriptional gene regulation. And in 2023 Catalan was given for development of effective mRNA vaccine against COVID-19. So here it was given for peripheral immune system, whereas microRNA we get in 2024 and vaccine technology we get in 2023. So these are the important, you should always remember the latest Nobel Prize winners and their achievements and their discovery. And the last question was about deficiency of which of the following leads to increased urine excretion of xanthurenic acid. So student, this is also very commonly asked and very important. So we know that if in a metabolism of tryptophan in kynurenine pathway, kynurenine can be converted to NAD, formation of vitamin B3 and xanthurenic acid. But for this conversion of kynurenine to NAD we require vitamin B6. And if this is deficient, what will happen? This pathway will be blocked and kynurenine will be diverted towards xanthurenic acid, lead to increased xanthurenic acid which will be excreted out in urine. So resulting in, so we can make out the diagnosis from increased xanthurenic acid level that patient is vitamin B6 deficient, and here it is due to deficiency of vitamin B6. So here you can see vitamin B6 deficiency leading to vitamin B3 deficiency, because from tryptophan niacin is not synthesized, because this require vitamin B6, the enzyme require vitamin B6, and in its deficiency most of the kynurenine is converted to xanthurenic acid and its level increases and started excreting out in urine. So the correct answer is pyridoxine, that is vitamin B6. So students, you can see there were many questions, there were many repetitions questions and there were new topics. So you should know that to solve INI-CET and get mixed marks, first of all you should know about all genetic techniques, and genetics is very important topic which is asked again and again. Second thing, you should know about sphingolipidosis and glycogen storage disorders and vitamins. So these are asked again and again. So here also we have questions about vitamin B6 deficiency, about how folic acid and vitamin B12 are interrelated. So these and thiamine is required for transketolase activity. So these are questions are asked again and again. Then you were asked about the concepts of metabolism of lipids and carbohydrates. Some questions were asked about proteins. Transport system is important. So these are the important questions and important topics which you should know by heart for getting good marks in INI-CET. I hope all of your paper must have went very good and you will get very good rank. Wishing you all the best for your result.